To the 8th module of this chapter. And what are we going to learn in this module? You're going to learn 2 new methods of asexual reproduction, spore formation and tissue culture. Let us go ahead and learn spore formation. Which are the organisms that exhibit spore formation? Rhizopus, or commonly something we refer to as the bread mold, the fungus that grows on the bread mold. Okay, these structures that you can see adjacent to me in the diagram, there are thread-like structures called as the hyphae. On the top of hyphae, there are some blobs. These blobs are called as sporangia. Students, focus on the word sporangia. Angia means an organ, and sporangia means organs that will produce spores. Now, how do these spores help in the reproduction of a new bread mold? Let's find out. During suitable conditions, these sporangia burst open and all the spores are released in the environment. Now, students, spores are going to help us in asexual reproduction. They're going to produce new plants, so they have to be well protected. And that's the reason they are covered with thick walls having multiple layers. Now, eventually, these spores, when they are released in the environment, they germinate. As you can see in the adjacent picture, the spore has germinated. When it falls on a suitable substratum, it germinates into a new bread mold and produces new fungal growth. This was spore formation. Moving ahead with a new technique of asexual reproduction, plant tissue culture. Students, this is an artificial method of asexual reproduction developed with the help of biotechnology. Let us find out how in an artificial method we can grow new plants. Okay, so in tissue culture, new plants grow by removing some tissues or separating some cells from the growing tip of a plant. Now the question arises, why only growing tip of a plant? Because the tips are rich in meristematic tissue. Last year in chapter number 6, the chapter of tissues, we learned that the tips of the plants are replenished or they are rich in meristematic tissues. Okay, after separating the cells, what are we going to do with them? The cells are placed in an artificial medium, usually a nutrient agar. The very word nutrient agar tells us that this is going to provide nutrition. Now, when the cells will receive nutrition, they will rapidly divide. As it is, these cells are meristematic cells. They inherently possess a capacity to divide and redivide. And plus, we are giving them nutrition, due to which they form new growth, large amount of cells. But these are undifferentiated cells called as the callus. Now, focus. We require a new plant and not just callus. So what do? We transfer this callus into a new medium, into another medium. Now what is special about this medium? This medium is rich in growth hormones, hormones like auxins, which will promote the stem growth and the root growth, gibberellins, which will develop the shoot system. And thus, the undifferentiated callus will grow into plant parts. It will start differentiating. Now, when they are started differentiating, they grow into new plantlets. But students, these plantlets can't keep growing in the test tubes. So what do? We transfer them. As they grow into mature plants, we transfer them into soil where they can further enhance their growth. Now the question is, tissue culture, can it take place in any atmosphere? Absolutely not. Students, using tissue culture, many plants are grown from one parent in a disease-free condition or in a sterile atmosphere. We are developing a new life. It has to be completely hygienic conditions. which are the plants that can be grown with tissue culture? Oh, this technique is commonly used for ornamental plants, plants which are used to beautify the landscape, plants which are used for our gardens to look beautiful. And in this way, we complete two new methods, spore formation and tissue culture.